Thank you for being here. So indeed, I'm going to talk about a tool which is called DiGeST and which allows to rank genes or variants in a distributed way using the Hadoop and Spark computing framework. So the design of this tool started with in the context of a bigger project called the DiGeST project, which ended up at the beginning of this year, and whose goal was to design a big data platform for storing and analyzing next generation sequencing data in the Brussels region. And this project involved 3 universities, the ULB, the UB, and the UCL, and 6 research groups. So 3 research groups were focused on IT issues and big data issues. These were the MLG, so we were coordinating the project, and the artificial intelligence groups of the UB and the ULB, and the 3 clinical partners working on different types of genetic diseases. So the motivation for that project was to, well, that DNA sequencing is becoming a routine in hospitals today, and so it generates a really huge amount of data. And so there is a need to share this data for 2 reasons. The first one is, well, to share the infrastructure so that it saves costs and money. And the other one is to have more data to be able to do some larger studies involving samples from different hospitals and study, for example, rare diseases, so for which hospitals, of course, have very few samples, and so to put all those samples together. So the project led to a number of nice achievements. So there were achievements in terms of, say, legal or ethical components like informed consent. So basically, where the patients allow research groups to share the data, to put them in a shared platform. Achievements in terms of databases, and especially a big database allowing to put genomic data and to share it among different labs. And then a series of tools ranging from bioinformatic pipelines for alignment, variant calling, up to high-level data tools for classification, for example, of variant data for different diseases. And so in this context, DiGeST is a tool that basically allows to rank genes or variants. Okay. So in this context, DiGeST, so the tool that I'm going to present here, allows to rank genes or variants according to their so how susceptible they have to be causative in a disease. Before going a bit further, I would like to know if everybody is familiar with what a variant is. Gene variant. If anybody is not familiar with what a variant, I just explained it.
Just explain 2 words.
Okay. So basically, with DNA sequencing, so we have most of our DNA is common, so we share most of it, and probably around well, less than 1% of it is different, and so that's what is interesting to look at to see what are the differences between 2 individuals. So here in DiGeST, we'll focus on case control approaches, where we have a set of people with a disease, a pathology, and a set of people, healthy people. And so we look at the like the variations in the genome between the case and the control groups. And so these variations are called the variants. And so we have about 3 billion base pairs in the DNA, but so we'll only store those like locations in the genome which are different from like the majority of the populations, so the variants. So we have this, say, this big database where we'll store all that information. So for one individual, I would have a set of a few millions entries for the variants in the genome. And here one line of this like big table represents a variant. So we have an ID for the patient it belongs to, and then the position, so that would be the chromosome position on that chromosome. Reference is what should be the letter at that location. Alternative is what is observed for that patient at that location. Zygosity is because we have pairs of chromosomes, so is the variant present on one chromosome, on both chromosomes, or of none of them. And then the gene symbol and some other annotations that we can add using annotation tools. So what's important to see here is that this table here can be very big. So currently, so following this DiGeST project, we have the data from the 1000 genome, which basically contain 2,500 genomes, and plus exomes from the different hospitals we were working with. So all in all, that's about 1 billion variants. And so in this table, we have the basic information for each variant plus annotation. So there is about 100 fields. So that's about 1 trillion entries in that database, so which is stored in the big data database for the project. And so from that, typically what we want to do is to compare two populations, as I was saying before, control 1 or the case 1. So it will mean to say which is the set of samples I want to retrieve from the database to create the case population. So set of sample ideas from that column here. Possibly a set of genes that I'm interested in. And then to create, well, to put that data into a format like this, a genotype matrix, where basically the rows are the variants and the columns are the samples. And the entries says what is the genotype of a given sample for a given variant. So here, for example, this means that sample 1 is heterozygous alternative for this variant, so which means that it has a zygosity 1, 0, only one variant on the pair of chromosomes. And from that genotype matrix, so here a set of samples are the control samples and the other one are the case samples. I will apply some scoring, so comparing the two populations, and then rank the variants or the gene according to that scoring. So that's the overall workflow of the tool.
Is the scoring based on the quality of the variant code, or is it based on what amounts to the code?
So the scoring is based. So you have different ways to implement it. I will start with an example. So a scoring for a gene. So this is what is called the burden score. So let's imagine that we have so these two groups: a case group with 3 samples and a control group with 4 samples. And on a given gene, I have 3 variants. And then I have that S1 has this variant, S3 has those 2, and is a homozygous alternative for the 3rd one. And in the control group, there is only 1 variant for sample S6. So, and then you have this 1-to-1 matching with the genotype matrix over there. And you have like say a nice general way to define a score is by saying that it's a weighted combination of the zygosity of the variants in your genotype matrix. So here, the load for a gene here and a given sample will be a weighted combination of the zygosity, so that's the 0, 1, or 2 over there. And here, if I put a weight of 1 for everybody, basically I'm just taking the sum for each sample for all the zygosity of the variants in that gene. So a bit more concretely, that would be here. This sum will be 1 because there is only 1 variant on 1 chromosome of the pair. If I go here, I would have so a score of 3, but then the standard approach is to set a score of 1 if there is at least 1 variant present in the gene. So here, because I have 2 variants, I still have a score of 1. And then in the control group, only sample S6 has a score of 1 for that gene. And then the score for the gene will be the difference between the score for the cases, so the sum of all those scores, and the sum and the score for the control, so the sum over all those scores, normalized by the size of the groups, and I take the difference. So here in that example, the score for that gene would be 2 over 3 minus 1 over 4, and so I would have this score in the end. And so then you can integrate in those scores notions on the data quality, for example, by using those weights here. Or it could be also what is the impact of that variant. So some variants are known to have some high impact in terms of pathogenicity. So then you would put a higher score for this one. So just a small thought about how much computing resources you need to do for that, since it's a talk about distributing computing. So the number of variants in that matrix can be on the order of hundreds of thousands, and the number of samples that you have can be on the order of, say, hundreds or thousands. So the number of entries that you have in the genotype matrix in the end is, say, only on the number of tens of millions. So given today's resources on computing machine, it's not so big, and so that can be run. This type of ranking can be run on a like standalone machine.
How do you compute the significance? Do you do some kind of permutation test?
So to compute the significance of this difference. So the basic approach is to use a Fisher test. Then there are other strategies. Actually, there is a really large literature on that. So that's we are using in the tool only the Fisher test to assess the significance of this difference.
Because I guess the significance takes more time to compute than the score itself.
If you take some say approaches which do permutation, yes, it could.
I guess if you go with fancy weights.
Yeah. So.
The Fisher test might not be the same.
So the idea is that in that if assessing the significance of the difference was computationally intensive, then you say the complexity here is on the order of the number of variants times the number of samples, then times a factor, and in that factor you would have the computational burden of the computation of the significance. So.
You could use also to accelerate non-parametric. I mean, if you need to make some permutation, the fact of implementing in a distributed manner would make it more realistic. So it's only just a reason why you tend to implement a distributed setting.
Just a factor.
But the reality is that doctors in that project, the only thing that carries the ranking. They want to see something they were not expecting. And it's more the ranking which is relevant for them because it's more a rare disease. It's not so all this p-value correction you typically have in gene expression here is less relevant. Also because there are so few samples, so few patients that any case you don't have enough samples to have significant. So most of the time it's just a way to suggest some strange variant.
But are you the believer of the ranking by p-value or ranking by
the ranking first in general, the p-value in this case it's more the effect size.
Yeah, it depends. I mean, yeah, the significance is also an important point. So yeah, there are different ways to assess it. Actually, the framework that we developed is quite oblivious to that because the way the computations will be distributed will make it possible to compute independently those significance on different nodes. So it will remain scalable, which is what we are looking for. And then there is also a lot of interest on like scoring not only genes but also pairs of genes or even larger combinations, because more and more people realize that diseases are complex and involve different genes, and that you have interactions between the genes. And so Then a possible extension on which we've been working is the computation of scores for pairs of genes. So here the idea is the same as before for basic scores, where for a gene, here we'll have a weighted combination of the zygosity of the genotype in both the case and control groups. And then for the score, we'll be able to bring together the scores computed for 2 different genes, and this for possibly all the pairs of genes. And of course, in this case, so here that would be an example. So for example, here we would have a score of 1, because so we put a score of 1 for a sample if there is at least 1 variant in both genes. And by adding, say, enlarging the set of genes which are considered, of course, there is an explosion of the number of combinations that have to be assessed. And here in this case, we end up really needed distributed computing to be able to compute all the possible pairs. Which brings us to TwoSparks, so which is the framework that we chose to do this distributed implementation, because that is probably the best framework for distributed computing now, in terms of trade-off between robustness to node failure in a cluster and the ability to perform high-level operations in an easy way. So it provides an API which allows to formulate a lot of complex problems like grouping, transforming, sorting data in a very transparent manner for the user. So which makes it easy and fast to implement solutions in a distributed manner. So that would be less efficient than MPI approaches for those of you who know about clustering, but the main advantage is that it's robust to a node failure. So how does it work, this kind of distributed implementation? I will take the example of counting the number of variants in a gene. So the input data would be the same as before, so each row codes for a variant, and so here inside that vector, so you'd have an identifier for a gene, then an identifier for a variant, and possibly other data. And the first thing to do would be to group the data by genes. And then for each group, you would count the number of variants you have in that group. And then you would use the sorting operation to sort all the results that you have. And you don't really have to care about who is going to do what in your cluster, because Spark will take care of dividing this matrix in blocks according to how you want to group data in that matrix. And then you will only have to design what you want to do for each block. So here it's just a simple counting, and then use the sort operation and get the result. It's almost as simple as that. So if we go for the scoring of genes, here instead, so that's exactly the same as before. So now we have, say, a bigger matrix with a set of capital G genes. That is exactly the same principle. And instead of counting the number of variants in a gene, I will apply a function, so this scoring function here for each of the gene keys, and then retrieve the results. So the complexity here is in terms of number of genes and number of samples. So just to put some ideas, if we have 20,000 genes and 1,000 samples, that's about 20 million scores to compute. And if we implement that and run on Spark cluster, so here we worked on the cluster that we have in our group, which is a cluster of 10 nodes, each of which has 24 cores, and 128 gigabytes of RAM, so that's 1.2 terabyte of RAM for the whole cluster, and there is 260 terabytes of data for storage. So I said before, it's computation is linear in the number of genes and in the number of samples. So here it's exactly what we see as expected. The colors is varying the number of cores, so how many processors you use to do the computation. So if we use one core, well, this is what we have, and the more core we use, the less time it takes. One thing to say is that here it doesn't take much time, so even for 30,000 genes, and in this case the number of samples is fixed to 100, it would take a bit more than 300 seconds, so which is about 5 minutes, to do all this computation with one core. So we could say it's a simple problem. And if we add some cores, so here if I go to 5 cores, then it's going to take about 1.5 minutes. So it's a gain of 3, factor 3, from going to 1 to 5 core. And if I go to 50 core, then it's a bit less than 1 minute, and the gain is clearly not 50, but much less, because there is all the overhead of communication in the cluster, which for a simple problem like this, well, makes the use of many cores not so useful. But what's interesting to see is that even for a simple problem, Spark manages to improve, well, to reduce the computation times quite significantly. And then if we go to the DiGeST ranking, so computing scores for pairs of genes, it looks a bit more loaded, but actually it's very simple to follow. So the first step is the grouping by gene, as for the monogenic ranking. And then we'll create for each group, so for each gene, a replication so that we'll have all the possible pairs in the set of genes. So here J1, from J1 we'll replicate the group J1 so that we have all the combinations with all the other genes, and we'll do the same for all the other ones. So at this step, from capital J gene, we'll go to all the possible pairs of capital J genes. So this increases the complexity to J to the square. And then once we have all those pairs of genes, we can compute the score and use the sort operation to retrieve the results.
The score is symmetrical, right?
Yes.
So you don't your confidence.
It could be, but it could be made so in this implementation it is symmetrical, but it could be made asymmetrical, because here so with the key J1, J2, we retrieve all the data from that genotype matrix that relate to J1 and J2. So you can decide how you combine the two.
It could be asymmetrical. Sorry?
It could be asymmetrical.
Yeah, it could be, but if it's not.
Yeah, I know. Yeah.
So here it's not computed actually twice, so you have just all the pairs. And so here this definitely increases the computational needs to compute all the combinations. So it's still linear in the number of samples, but quadratic in the number of genes, and so that's what we observe also experimentally. So here, if we vary the number of genes, we see that the increase in computation time is below this quadratic increase. But if we increase the number of samples or a number of genes fixed, then we have this linear trend. And so here, what's nice to emphasize is, so computing the scores for all the pairs of genes with 30,000 genes would take about that would be about 1.5 days over there using just one core. And if we go up to 5 core, then it goes down to about 8 hours. And there is almost a gain of 5 here in terms of computation time, which means that Spark almost perfectly distributed the computation, and the gain is as much as the number of cores that we added. And then if we go and increase the number of cores, the gains are less proportional to the number of cores that we add because of the overhead of communications in the network. But here for 50 cores, for example, the gain is about 25 times in terms of computation time. It takes a bit less than 2 hours in this case. there are any questions? And so then we implemented all this, so both on a cluster backend and a web front end, which can be which is available from the web. Well, basically from the web interface, the user can design his control and case groups with different filters that can be applied on the variant database. Then the filtering, creation of the genotype matrix, and the application of the scoring function would take place on the cluster backend. That would return to the web interface the scoring. So a few snapshots on the interface. So here is the widget that you have to create the filter. So you can select the variable and then some operator and the values you want to associate to that variable. So for example, take a subset of samples, then for which the quality is good enough, and then which have, say, an impact in terms of a functionality which is high, and then you can save filters for retrieving them later, and that would allow you to create the groups of well, the case and control groups that you want to use in your analysis. Then to start an analysis, well, you specify the control group and case group that you created before, the scale at which you want to perform the scoring, and then give your analysis a name. And then you retrieve in the scoring tab the ranking. So here it's an example for a diagenic ranking. So you have the set of here 2 genes which are ranked first, and then some details about the statistics, how many times variants were seen in that pair of genes for the set of samples that you were studying. And so that's about it for the tool. So the key takeaway for me is that so it was not sure at all what we would find in terms of experimental results with like using Spark for this implementation. And it was surprising to see how efficient the framework is able to distribute the computations, even for simple problems. Then for future work, so we're working on the integration of this tool into standard bioinformatic pipelines like P-Link or Bioconductor and workflow tools like Galaxy, and going towards scoring of like larger combinations of genes as well. That's it.
Bottleneck, the number of variants, the number of patients, of genomes. What is, I mean, the variable or the dimension that slow down the most this kind of analysis?
The CPU speed. No, that would be the how many genes you, well, the combinations of genes, of course, because this really will make the number of combinations explode.
So if you want to hygienic,
yeah, yeah, that's that would be.
So it's the variable which makes the
but you could have as many people in your database without slowing down.
Yeah, it's like, I mean, one idea would be, for instance, to extend this digest to another node, because at the end it's the idea of taking a couple of features and doing something. Then it could be some mutual information. And so this is indeed the number of variables and how large is the dimensionality of the subset that you consider to compute the score. Because if it's a univariate, it scales linearly, but then quadratically.
So how many times in biomedical science we leverage co-occurrence of variants instead of just single variants? I know tons of cases of single variants, but how many times we found that those genes alone that do nothing is really when they co-occur that they do something? You know, it's a good idea, but it's exploding your CPU. And
it's really research of ID squared that is common enough to work with. So diagenic is really one of the pillars of the relation of squared.
So
and there is a database that they need where there are a lot of digenic disease, and they make a database data where they're really collecting a list of diseases for which people found evidence of diagenic interaction. There are enough.
Yeah,
This
for
is
the
really
moment
next
that
frontier, because they realize that with only GWAS taking one variable at a time, they are not able to discriminate as expected. And so the only ways to go up in the dimensionality is move to diagenic and then move to oligogenic. So this is really the frontier of genetics. Now, as you said, the reality that it's much so expensive, and then probably you don't have enough statistical power for doing that. But I think in GWAS you have, because in GWAS they start having 100,000 patients. And so even if you have million variants, you can have statistical stuff.
And why not do the network approaches for that kind of stuff? We focus on bivariate.
No, I mean, this is the start. I mean, this again is a feature selection implemented this part.
To do a MRNF on the conference.
Yeah, one score could be a bivariate mutual exclusion. So you could use redundancy and you could use glycogen.
so that in this database, so it's called DIDA, and it's a database in which people can contribute as well. But for the moment, there is about 200 diseases.
This network is not mainstream. In all these genetic studies, there are always association studies. So, and most of the time they are univariate. So again, people make univariate tests like odds ratio. It's actually the world of odds ratio. And already moving to bivariate, it's a big step. And network is at least the mainstream research is not coming. Yeah. But I mean, why not? It makes sense to have network.
But in genetics, they can compute a percentage of variance explained by a single variant.
Yeah.
I'm wondering if what's the
now realize that is not enough. I mean, they're not explaining enough variation in single variant.
Your variants are in the whole genome or only exomes?
So for the data that we have from the clinical partners who we're working with, it's only exome data. And otherwise, we integrated the data from the 1000 genome, and so there is genome data.
Just I'm curious about research. Yeah, how many, like every individual has how many SNPs?
So in terms of exomes, it's about 100,000. So for one exome, there is about 100,000 SNPs, and for one genome, it's about 3-4 million SNPs.